Before you can make your own standard curve, it helps to know what a standard curve looks like. So here's one for hemoglobin, the protein hemoglobin. We're looking at absorbance on one axis and concentration of this hemoglobin standard on the other axis. And then we have an example showing cobalt chloride. Again, look, here's the line we get looking at absorbance versus concentration. And finally, looking at bacteria and how they absorb when we look at them in a spectrophotometer. So what do these graphs have in common? Well, most importantly, we're looking at the relationship between absorbance and the concentration. In other words, the amount of material, whether it be cells, protein, or a chemical. In class, you used your spot plate data and absorbance, and now we're going to create the standard curve using those two. So first we're going to look at the number of colonies from the simple dilution, and that's shown in the blue box here. Now the problem with this is that there are no isolated colonies in any of those simple dilutions. Each spot has too many to count. These are all masses of individual colonies that have grown together. There were no colonies from the simple dilutions. There were too many to count on the spot plate. So how can you complete this table? How do you determine the number of cells in 1 ml? And this will become important because you're going to use data from this table for your standard curve. Since you used a single yeast culture to make both the simple and the serial dilutions, we can count colonies on the spot plate from the serial dilutions and use that number to calculate cells per mil for the original culture. So let's look at those serial dilution colonies. You can see that at 1, 2, 3, 4, at the 5th dilution, we actually can see isolated colonies. And if I count those, I see that it looks like there are about 3 colonies on the top and maybe 4 to 5 in the bottom. So let's say that the average is 4. Here we took the number of colonies that we counted on spot number 5 and plugged it into this table. So if we had 4 at that dilution, we had 40 here, 400, 4,000, 40,000, etc. And then we can go down and determine if there were 4 cells in 3 microliters. We divide 4 by 3 to get, we're going to round it to 1 cell in 1 microliter. And then to go from microliters to milliliters, we're going to multiply by 1,000 because 1 milliliter contains 1,000 microliters. So we now have 1,000, 10,000, etc. And we now know that our original culture contained 1 times 10 to the 7 cells per mil. Now we're going to transfer this number to table 1. So we're now on the simple dilution table. Remember, we couldn't determine the number of cells per mil because there were too many to count, but we've taken that number from our serial dilutions. And now we're going to be able to, knowing the number of cells per mil, we'll be able to basically calculate forward and determine how many cells per mil were in each dilution by starting with 1 times 10 to the 7 and using the dilution factor that we calculated for each of those tubes. Now we've filled in all these numbers And just in case you forgot, we started with 1 times 10 to the 7. And if we've got a dilution factor of 1 to 20, which we derived from our volume of yeast, 50 over the total, which was 1,000 microliters, because it's 950 plus 50. That's how we get it 1 to 20. And to get to 5 times 10 to the 5, we're going to divide 1 times 10 to the 7 by 20. That's going to give us 0.05 times 10 to the 7, and we simplify that to 5 times 10 to the 5. Now your job is to graph these values against your absorbance readings for the simple dilutions. And I'm not showing them here because I want you to turn in your own graph with your own absorbance readings. But when you're done, your graph should look something like this. Here's our standard curve at Saccharomyces cerevisiae. We read absorbance at 600 nanometers. Here are the absorbance readings up to about 2.5. Those were values that I came up with. And then here are my cell concentrations that we determined and plotted from the previous table. Why is it useful to have a standard curve? Well, let's say you come in on a Friday morning, it's 8 o'clock, and you need to be in your class by 8:30, but you also need to read and determine the concentration of your cell culture that's been growing overnight. So you can turn on the spectrophotometer and set the wavelength to 600. You can transfer 1 mil from your culture tube into a cuvette. You set the instrument to 0 with water, and then you read your culture, your sample of yeast, and you find out that the absorbance is 0.86. Now, because you have previously created a standard curve for yeast, you can calculate cells per mil in your tube. Here's a graph that we've used for that. It's slightly different than the one that you've seen. And again, we have our absorbance of 0.86, and we're going to try to figure out how to use our graph to take that number and convert it to cells per mil. So what we're going to do is find out where that maps on our x-axis, 0.86, because that was an absorbance reading. So here's 0.86. We can then go straight up and see where that point intersects our line. And this is the line that you determined from your cell concentration from your spot plate versus absorbance of the samples that you did in class. Here are those particular points, and then we simply connected them with this line. Now that we know where it intersects the line, we can go straight across to the y-axis and find that we've got 1,200, and that's times 10 to the 5th, because we didn't want to put all those zeros here, so we simply write it on the legend. And that means that we have 1,200 times 10 to the 5, or we'll simplify that to 1.2, times 10 to the 8 cells per mil. The other thing that's nice about these graphs is if you do them in Excel or another similar program, they will be able to calculate the slope of this line and give you a formula, and with that, to determine what your y-axis value is, all you need to do is plug in the 0.86, and that's shown over here. So here's the calculation for the slope of that line. And now I've simply taken those numbers down times 0.86 to get this value, and it gives me 1,201, very close to what I manually measured.